So as I said, the score on the exam was expected. When you have a certain set of behaviors, if you read the handout that I gave you, if you read it, I printed it, it's on the door, it's at the door as you walk in. I sent you the color version. I think you'll see that I had expected these results from you. This was not a surprise. When you're late, consistently late, when you're not paying attention when you are here, when your notes are not complete, that's a problem. That's already you're one step behind. Then on top of that, you add the difficulty of reading a question and then applying the knowledge. So let's go through each of these questions today. Identify where the answers came from. Where did we cover this? And how do you answer a question like this? Then at the end of this, once we're done doing an item analysis, this is what this is called, an item analysis, you're going to get a second chance to do the test. It'll be a different test. And whichever test you score higher on, that's the one I'm recording in the grade book. We're done after that. So we don't have a lot of time because today's a shortened day. So we're going to move on through this. So which of these often serve as receptors or cell recognition molecules on cell surfaces? I told you when we went over the extracellular matrix that glycoproteins Oh, I apologize. Give me a second.
Can I turn off the light?
Yes, please do. Glycoproteins were used as ID molecules. I spent an entire day talking about the extracellular matrix, talking about the idea they have these proteins that then have attached to them sugars. You having conversations while I'm explaining something is part of the problem. There's many facets to the issue of not doing well on an exam. There's a lot of different reasons why. Certainly part of it is that there's a lot of reading on the test. But another part of it is that you guys, as I keep saying, some of you don't listen. And it could be for various reasons. And some of you said it was because you were sick of hearing me talk. So I gave you the amoeba sisters in a virtual lab to do. And still nothing happened. So how do we solve the problem? That's what that flyer's for. That's why I communicated that to your parents and to you. Your parents have a copy of that as well. So glycoproteins are proteins, and this is the protein part here. And you can go back and look at the video on extracellular matrix, and you can see that I mentioned this several times and tell you it's going to be on the test, with a sugar. And the sugars attached to it are called glyco. That's what glyco means, short for like glycogen, right? Glycoprotein. Glycoproteins are things that attach to the outside of the cell membrane that identify this cell as being part of you. Are we clear on this? There should be no surprise here. There should be absolutely no surprise at this. It's on the board. And you guys, anything I don't say you don't understand, go back and look at the review video where I tell you it's in the review. It's in the video in the class notes. I gave you this in class notes. And you can go back and look at it. If you didn't write it down, then that goes back to the whole idea of making sure you do the first of the 6 R's, which is record the information correctly, which you can't do if you're on your cell phone or if you're giggling and talking and whatever. Organelles other than the nucleus contain DNA. What are the 2 organelles that we talked about that contain their own DNA? Yeah.
Mitochondria and chlorophyll.
That's right. Mitochondria, which look like something like this, double membrane. We talked about what those organelles were, what they did, what are their shapes, where they, and we said there was going to be some There was some foreshadowing of what's coming with the mitochondria and chloroplast when we get into cell energetics, but we have mitochondria and chloroplast. So you do have to know what they do as well, I said. In fact, you had an entire activity, podal activity, where you had to identify what the structure and function of each organelle, and this was one of those things. They said mitochondria have their own DNA, chloroplasts have their own DNA, and the answer was mitochondria and chloroplasts for this question. The liver is involved in detoxification of many poisons and drugs. Which of the following structures is primarily involved in this process and therefore in abundance in this liver cells? Well, what is the answer to that one? Okay, you said transport vesicles. Well, the answer is smooth ER. The smooth ER has two functions. Again, part of that activity that you were given and stuff that we talked about when we talked about the endoplasmic reticulum. The smooth ER detoxifies things that you eat, any poisons you eat. The smooth endoplasmic reticulum uses enzymes to make them not poisonous. And the second thing that the smooth ER does is it makes lipids. It doesn't make proteins, it makes lipids. And there's also some modification to proteins. So when we talked about the secretory pathway, what was the pathway of proteins leaving the cell? We talked about the function of the endoplasmic reticulum. I showed you a three-dimensional movie on it. We walked through it. We talked about the smooth ER. These are really key points. Again, taking pictures is fine, but actually listening and writing it down, thinking and asking questions is a lot more important. I think if you keep doing the things you're doing before the test, now I think you're going to get the same results. In fact, if I remember correctly, the definition of insanity is if you keep doing the same thing over and over again and expecting something to change. That is literally a definition of insanity. Yeah. Endoplasmic reticulum. Yeah.
What's nuclear envelope?
A nuclear envelope is the nuclear membrane. It's another word for the nuclear membrane. So it envelopes. An envelope is a surrounding. Guys, taking pictures of the screen when I'm posting it online, and when you have a lot of this already in your notes, at least online, in Schoology, I just think is not productive. Writing it down with a pencil is much more useful. Nuclear envelope is another word for membrane. Nuclear membrane. Envelope means to surround. That's why to envelop means to surround.
What do the rougher ER use?
What? That's something you should know already, right? What does anybody know what the rough ER does? It has ribosomes. That's why it's rough. And what do ribosomes do? No. So, my children, this is why we didn't do well on our exam, right? Because these were the functions that you needed to know. The organelle functions. Ribosomes are an organelle. They make proteins. The rough ER makes proteins that are what? The rough ER makes proteins that do what? Secretory pathway. They're going to leave the cell. The rough ER makes proteins that leave the cell. That's it. The ribosomes in the cytoplasm, the free ribosomes, not on the ER, make proteins that do what? That's right. Stay in the cell. We talked about it. We looked at it. We watched a movie that said this is how it works, and you actually saw the protein being made. I said you have to know this. I literally said you have to know this. It's going to be on the test. When I said that, I saw no one writing it down, but I did see people taking pictures of the screen. I did. I saw people taking pictures. I remember this particular moment. And I remember thinking to myself, when they come to the test, they're not going to get the question right. Because I don't think they're processing it. They're not asking any questions. They're not writing it down. They're probably not making index cards for this. I don't think they're going to get that question right. And now today, after the test, people are asking me, what's the RER? What do the ribosomes, you don't know what ribosomes do. Now this is the problem. Are we clear? Yeah. It's not the test. It is not the test. It is how well you're preparing for the test. And that's what we have to grapple with. All right, let's take a look at number 4. The patient has had some serious accident and lost a lot of blood. By the way, this came right out of that Amoeba Sister video. She used this exact example. I could play it for you again, but I won't. I'll tell you to go watch it again in Schoology. And if you watch the video, they literally use this exact example. The patient has had serious accident, lost a lot of blood. In an attempt to replenish the bodily fluids, you put equal amount of water, blood lost, you replace it with pure water. Distilled water means pure water. That means no solute, right? No solute, 100% water. Into the veins. What will be the most probable result? So if you have a cell and you put the cell in 100% water, you know the inside is not 100%. Whatever it is, it's not 100% water. So this is higher water concentration. This would be called hypotonic because it has less solute in it, hypotonic. That means water is going to go into the cell. Water is going to go into the cell. Water does move when the solute can't. Water goes into the cell. The cell will swell up over time and could, in fact,
Burst, can it?
that's right, burst. Just blow up like a balloon that's overfilled. So that's the answer to that one, and that answer is different for each one, but there it is. That's the answer. Hypotonic, hypertonic, isotonic. That's what I wanted to cover today. That was the plan. That's what was on the syllabus. But I did have this as an alternate plan if my suspicions were correct and you were not going to do well on your test. I wanted to be ready to go over this. That's why I gave you that flyer. But hypo means under, and tonic means solute. So low solute. As compared to what? I would ask you to watch the video.
Hypertonic.
As compared to hypotonic, this is hypotonic as compared to what's in the cell. The stuff in the cell is hypertonic as compared to what? The stuff outside. Everything, hypotonic is a relative measurement. Low and high is relative. When you say this is a hot day, that's relative. Relative to what? To something that's colder. All right? So it's not an exact measurement. Hypertonic means high. Hyper means high, solute. And iso means the same. These are words you have to know for sure. And we'll talk more about this. Same as what? Same as the inside versus the outside. So just like the syllabus says, we're going to do what we can today. Tomorrow we'll do more. And I'll keep doing this whole video today. Wherever we stop, I'll pick up with the other class. And I'll post it. And over the weekend, I expect you guys are going to watch it all and go over all these questions. And then on Monday, we're going to start with going over solute, solvent, solution, hypertonic, hypotonic, isotonic, what happens to water, osmosis, when do you get osmosis, when do you get diffusion, and hopefully when you retake this test, it's an easy A because you prepared correctly. All tests is less a measurement of your intelligence and more a measurement of how well you prepared. I hope that makes sense. Number 5, 6, 7, 8, and 9 simply had a really detailed, I think beautiful, it's right out of your book. Your book has it. I used the same picture in class along with some sketches, and I even drew it out. I think I did something like a matrix that looks like this. I did in class, and then you had the membrane, which had your lipid layer, and then you had some proteins in. I drew some proteins in there. You can go look at the video, and something like that. And then you see, and I told you cholesterol. I said cholesterol is in the membrane. It helps keep. It's only found in animal membranes, and I said cholesterol. It helps keep the membrane fluid in the winter, so you have more cholesterol in the membrane than you would at other times during the season. And I said, when you look at, and this is one of the things I talked to you about, is that you can have glycoproteins. This is when I mentioned glycoproteins as tags on the outside of the cell. But you could also have glycolipids. And I said, a glycolipid is just instead of a sugar being attached to a protein, the sugar is attached to the lipid. If you look, you'll see that there's a hexagon, a series of sugars that are connected to a lipid. Do you see them? It's B for everybody. So you see that cholesterol is that little molecule that's in. So cholesterol is here in the membrane, so that would be E. Now, the peripheral protein is the protein that's embedded in the membrane. So peripheral means the outside of the cell. So that's that big hunk of thing that's sitting there. That would be D. And fiber in the extracellular matrix is the stuff that's outside the cell. The only thing that's outside that's not attached to the cell is sticking outside the cell. It's A. C is inside the cell. In here, you have the microfilaments and microtubules of the cytoskeleton. Remember, we looked at that in the video, and we went all the way down to the nucleus. We said the inside has all these. The skeleton, then it's all attached to the inner layer. Remember, we actually look, and the video is still posted on Schoology. It's that tour of the inside of the cell. Yeah. No, but at the end of the day, if somebody wants a copy of them, you can come and get a copy. Five is E, yeah, and six would be D, and 7 would be B, and 8 would be A, and 9 would be C. So 9. This is inside the cell is the cytoskeleton. Outside the cell is the extracellular matrix. The glycoproteins are sugars. Again, remember, sugars look like this. We draw sugars like this. We talked about it. We draw sugars like that. If you see a hexagon attached to a protein, that's a glycoprotein. The hexagon's attached to a lipid is a glycolipid. All right. Which of the following produces and modifies polysaccharides that will be secreted? Secreted. Well, it's not a vacuole. They don't modify anything unless they're digesting it. Mitochondria don't modify anything. They make ATP. They make energy for the cell. The lysosome also, like the peroxisome, both of those are used to destroy cells. Destroy macromolecules. They break down macromolecules, right? So the only one that's involved in the secretory pathway, if you learn the secretory pathway, which we spent a whole period doing, and there's a video posted in the exam review. There's 2 videos, one called Exam Review Part 1, the other Exam Review Part 2. We did it in class, posted in Schoology. Golgi body is the answer. Answer 10 is Golgi body. 11. Bacterial cells' DNA is found in its... I knew people would say nucleus. I actually told you, I literally said, I wish I could had time to edit videos and just play little snippets of me telling you. I literally said when we covered prokaryote versus eukaryote, I said prokaryotes don't have a nucleus, and even though I'm telling you this, somebody in this room is going to put nucleus on the test. I said, even though I'm telling you this, I said, write it down, put it on an index card, memorize it, prokaryotes have no nucleus. So it says bacteria. Bacteria is a prokaryote. There is no nucleus, so it can't be A. There is no DNA in ribosomes. Ribosomes are just RNA and some protein. Capsule, something completely not, it's made of protein, but it's not anything you need to worry about right now. Peroxisomes are vesicles that contain peroxide that destroy things. It's and by the way, nucleus, ribosome, nucleus is a membrane-bound organelle. Peroxisome is a membrane-bound organelle. Bacteria, prokaryotes, they have no membrane-bound organelles. It's part of the qualities that you needed to know for the test. I said, look, prokaryotes. Again, in the exam review video, you can go and watch it. It says prokaryotes, there's 2 kinds. There's bacteria and there's archaebacteria. I said, in both of these, no nucleus. They have no membrane-bound organelles. It's in your chapter reading. It's in the activity I gave you that looked at prokaryotes versus eukaryotes. It's in the class notes. It's in the class test review. And they don't have a nucleus, but they do have a nucleoid. And I actually drew it. Nucleoid. And I wrote the word nucleoid region. Again, knowing it was going to be on the test. There was no doubt. And I warned you that somebody was going to make that mistake. And I hoped it wasn't going to be you, but obviously it was most of you. A child brought to the hospital with a fever of 107 degrees Fahrenheit. Doctors immediately order an ice bath. I literally used this exact example in class. I explained it to you. I told you I was going to ask you a question on it. I went through it exactly in class. I told you a story. That's one of those stories that you tell me you don't want me to say to you, that you just want to watch the video, you just want the facts. But this was one of those stories I tried to explain to you that this is what happens in hospitals.
Don't the enzymes go
Excellent. That's exactly right. The enzymes, when it gets too hot, so what happens to proteins, any protein really, but enzymes especially, enzymes are proteins mostly, some are RNA, but mostly proteins. What happens when they get too hot? When it gets too acidic? I think we even did a lab on this.
Yeah.
Too acidic, that's right, acidic. When it gets too basic, right? When it gets too salty, any of this stuff, any of these things get too much. That's what up arrow means, too high or too low, either way. Outside its normal range. The protein the protein denatures. That means it goes from its normal shape, let's draw it like Pac-Man, to some kind of gobbledygook. It stops working. But when you bring down the temperature, you bring down the acidity, you bring down the whatever, if it's too basic or too salty, get it back to its normal range, it goes back to its normal shape and starts functioning again. This is denature when it stops being its natural form. This is renature when it goes back to its natural form. This is the natural form, the way it's found in a living creature. So that when the body, elevated body temperature denatures the enzymes, that would interfere with the cell's abilities to catalyze various reactions. That's what enzymes do. They catalyze reactions. They make reactions happen. And they can't do it if they're denatured. And that would be the answer to that one. All right, moving on to 13. Now we're already more than halfway through the test. I think you see these are not that hard as long as you knew what we were covering in class. Under which of the following conditions would you expect to find a cell with the predominance of free ribosomes? What do free ribosomes make? Proteins that do what?
That leave the cell. That stay in the cell.
Stay in the cell. The free ribosomes make proteins that stay in the cell. So cytoplasmic enzymes, A would be the answer to that one. Extracellular matrix, that's outside. Enlarging its vacuole, I'm not sure how vacuoles are made of phospholipids. Making a protein would not enlarge that vacuole. Digesting food particles, not sure. They would make enzymes that are used to digest food particles, so in an indirect way, it's not the best answer. Secreting proteins would be in the ER. So the answer is the cytoplasmic enzymes. Cytoplasmic enzymes. Now, even though you're going to get a retest on this exam, you understand I'm changing the questions. So you have to know the topic. I'm not going to try to make it harder, but I have to change it. So large numbers of ribosomes are present in cells that specialize in producing which of the following molecules? Proteins. That's easy. You know that more than half the people got that wrong? Ribosomes make proteins. It's just simple. 16, small nonpolar hydrophobic molecules such as small nonpolar hydrophobic molecules pass through the membrane easily. Period.
We skipped 15.
Oh, what was 15?
The child dies.
Oh yeah, child dies. So when we look at a child dying in that example, this happens, by the way. It's sad when it happens. But there's a bacterial infection that's infecting the child. It could be any kind of bacteria. They do an autopsy and they find that the white blood cells have the white blood cells are supposed to eat it. Remember I showed you white blood cells eating bacteria? Yeah. Said this is how it's supposed to happen. They surround the bacteria with these, and they fuse them with the lysosomes. The lysosomes are supposed to kill the bacteria. Right? That's what's supposed to happen. But when they opened up the baby's cells and looked under a microscope, they found out that the bacteria were eaten by the white blood cells, but they weren't digested. So let's look at the answer options. Surface receptors of white blood cells permitted bacteria to enter the cells. So the surface bacteria, the surface proteins, receptors of the white blood cell did not allow. There was a defect. Is that right? Yeah. Look at it. There was a defect in the cell surface of the white blood cell that did not allow the bacteria to get engulfed, to get swallowed, to get taken in. Is that right? No, that's not right, because the bacteria were already in the cell. So it's not that one. It would be B. The defect in lysosomes of white blood cells prevented the cells from being digested, from destroying the engulfed cells. These cells are engulfed, they were swallowed, but they were not digested. So it is B. And the rest of these are wrong for other reasons, right?
Yes.
Okay. Are we good? Do you want me to go through the reasons why they're wrong? If you should read them, though, when you come and get a copy of the test. You should look at that. Whenever you're looking at a test, an old exam question to study, you have to ask yourself, why are these other ones wrong? Because the next question you get on the next test will be different. All right. Small nonpolar hydrophobic molecules pass through the membrane easily. So let's just be clear on that. 16. If there's anything that's small gets through relatively easily unless it's charged, especially if it's nonpolar. And hydrophobic means nonpolar, or nonpolar means hydrophobic. So small nonpolar molecules get through the membrane easily. Done. That's there's just nothing else to that. 17. Which of the easily depending on which test you have, actively transported across the cell membranes would be wrong. It's
the passive process, like it go from a higher region to a lower concentration.
Easily pass through the membrane's lipid bilayer. Actively transported across the cell membranes. Active means something very specific. You need energy to do it.
So that thing, it has to be charged, right?
Charged ones need active transport. That's correct. Or it could be passive, but it need facilitate. We'll talk about it next week. Which of the following statements is correct about diffusion? Well, what's correct about diffusion is what for 17? Everybody should know this. This was a simple. Oh, you just said it. I'm sorry. I thought you were talking about 16. I was looking for that answer. But yeah, that's right. 17 is real simple. The passive process in which molecules move across. The others were silly.
Yes.
So it's A. 18. Which figure, there's a bunch of figures there. Which figure is an animal cell placed in a solution that's hypotonic? Now this is just a repeat of a previous question. I think one of the first two questions was just basically the same question. You put a cell in a hypotonic, what happens? Now, which of the three are animal cells? The ones in the bottom are plant cells.
So
They have cell walls.
A, B, and C.
A, B, and C are animal. In fact, they're red blood cells. We drew them in class when we talked about sickle cells. But yeah, they're red blood cells, animal cells specifically. The middle one, B, is isotonic. See water going in and out? It's dynamic equilibrium. Water's going in and out without any change, any net change. C, water's leaving so much that it actually shrinks too much and the cell dies. It's called plasmolysis. And A, it blew up. Too much water went in. All right. So A is the answer. Cell A, which is different for each of you. But cell A is the answer. White blood cells engulf bacteria through what process? How did the white blood cell in number 15 get the bacteria? And when I showed you that, when I drew it on the board and then showed it to you in an animated video, I used the word phagocytosis. And I got a bunch of giggles for some really ridiculous reason. So it's phagocytosis. It's the swallowing. Phago means swallowing. Cytosis means cell. So it's large. It's taking a large amount of material. Obviously, if you're taking a whole bacteria, that required that. I drew it out for you. Remember, I drew the endocytosis, the cell membrane, literally forming around the bacteria, and I drew it like 3 times or something like that, and I showed you it was over time. Then we watched a video on white blood cells doing that. All right, 20. Which of the following is a reasonable explanation for why unsaturated fatty acids help keep any membrane more fluid at lower temperatures? I have said this multiple times. We talked about phospholipids. They could have fatty acids that are saturated. They look like this. or they could have a fatty acid where one's saturated and the other one has a double bond, which makes it bend. That bending is important in part because it makes more space between the molecules. Huh?
Oh, never mind.
More. This is the phosphate, and these are the lipids.
No, I
No,
know that
on
is, I mean,
above
like
more.
under the yellow, but I realized it's just an arrow saying more space.
Yeah. My bad. Let me redraw that then. I'll need her. There's more space in between the phospholipids because one of the fatty acids and the two fatty acids in the tail, one of them has a double bond. It bends, and there's more space in between them. And that's the answer to that one. It's the unsaturated fatty acids. The double bonds result in let me see. Double bonds form kinks in the fatty acid tails, forcing adjacent lipids to be further apart. Which is one of the main energy transformers of the cell? And here's again, this is the fifth question or sixth question about the function of the organelles. You had a whole activity in class. You were to turn it in for homework. We covered them in class. I mean, wow. And I spent a whole, I want to say a third of a day on mitochondria and chloroplast. And we'll go over it again, but when we do energy transformations, but mitochondria is responsible for energy transformations in the cell. The lysosomes digest things. The peroxisomes help break things down as well. The Golgi apparatus packages things for leaving or for going into different organelles. The vacuole holds things. It could hold food. It could be a food vacuole. It could be any kind of material. Where the mitochondrion contains the resources necessary to make energy and transform energy, change energy. Celery stalks are immersed in fresh water, and one's immersed in salt water. So again, what I tell you when you're working with these questions is to go ahead and do a drawing. You put a celery stalk, which is cells, they're plant cells. You put them in water, where you put one is hypotonic, and the other's hypertonic. Hypertonic means high salt. That means water's leaving. Hypotonic is high water, low salt, so water's going in. So the answer to this question is the reason that this one wilts. I actually used the word wilts. I drew a flower on the board. You all laughed at my flower. Wilts. And this one is stiff, crispy, and straight. This is why plants wilt. They wilt when there's too much salt because the water gets drawn out. Osmosis occurs. So, and for there you can look at hypertonic in both, hypotonic in fresh water, but hypertonic in salt water, which causes the salt water one to shrink. And I should label this salt water, although you should know. And this one being fresh water. And I think the amoeba sisters talked about the fish being in salt water versus fresh water. That's right. But I discussed plants, and I drew a flower for you. So this question was clearly in two different ways you got this information. 23, they had 5 dialysis bags. There was a graph of what happens. The dialysis bags, again, are semi-permeable membranes. There's a paragraph that explained all this. And you put these bags in 0.06 molar solution. And one of the bags increases water weight, and therefore water a lot. One increases a little bit less. One doesn't change at all. One decreases mass a little bit. And one decreases mass a lot until it reaches dynamic equilibrium. So there's A, and then there's B, and there's C, and D, and E. This was the same for all of you, and as far as the answer choices. Why would these two gain mass?
Because they were put into a saltier,
Not salty. You reversed it, but you're right. It's These were put into hypotonic solutions. Why would the other ones lose water mass?
they're put in a hyper.
Because they were put in a hypertonic solution. So these lost mass because they were put in a hypertonic solution. And obviously, the one E is the most hypertonic, A is the most hypotonic, and what was C?
It was basically the control.
It was isotonic.
It was the control. You're right, but it's also isotonic, or the same, so it didn't change. So you're right. In both cases, it was the control and it was an isotonic. So that's how you need to read those. That's how you would have read that chart if you would have known what the Amoeba Sisters try to tell you, what I went over in your review on our virtual day. Hypotonic means low salt. Hypertonic means high salt. High salt water gets drawn out. Low salt water goes in. All right. 24 says, which structure the site of the synthesis of proteins that may be exported from the cell? This is the third question on the secretory pathway. So which one is going to make the proteins where the proteins are leaving the cell? Where are the proteins made that are going to be leaving the cell? The mucus, the spit, the sweat. It does go through the Golgi before it leaves the cell, but that's not where it's made. Where are the proteins made? What's the term? Rough ER. The rough ER. It's the rough ER where the proteins are made. Then they go into the smooth ER. They get changed a little bit. They go into a vesicle. From the vesicle, they go into the Golgi. So you're right. Well, I already said it. You drew it. It's drawn. It's in the notes. I'm just reviewing it. I go over it in tutoring if you want, but we don't have time. I wish I could. This is not the first time you've heard this. From the vesicle, goes into the Golgi body, gets changed again, goes into another vesicle, gets transferred. Remember, I showed you it being walked to the cell membrane where it leaves the cell. That's the secretory pathway. Which of the following contains hydrolytic enzymes? Enzymes that do hydrolysis. That's what hydrolytic means. Hydrolysis. Enzymes that break things down. Which of these has those? Lysosomes have those. That's right. Lysosomes. B. That's the one you learned about. Now the extra credit. That's the whole test. That's it. So maybe you had a rough time with a couple of them, but I don't see why anybody could. Can anyone argue that you could not have done well on this test? Like, is there anything in those questions, on the basic questions, that we did not cover? All right. You can come and get them at the end of the day, but I need them for the rest of the classes. So hand these back. Come and get them, because they're useless to me because we already did the test, but we'll do it again. All right, so I hope you guys understand we can do better. We can do better on our next exam. There should be hope.